Hello? Hi. Hi, how are you? All right. Thank you. Yeah. Yeah, we'll see. Um, you know, the best way to do it is go back and look at the recordings from, uh, past and see what we've been doing. But yeah, we also have our repositories that, you can look through and see if they're, I guess in the DevoLearn organization, there's some open issues. So you can check those out. Yeah, that's good. Okay. All right, so that's good. Welcome to the meeting. I don't know. There's Morgan. So I think this is going to be the last meeting of the year. We had a pretty good year. We did a lot of different types of activities in our meetings. We had talked about a lot of different topics. Of course, we had our summer of code students, and they did their projects. And we had a couple of weeks where they presented on things. So we're working on a paper for the DevoTG. So that's coming along, actually. That's Jayadratha and myself. That's coming along pretty well. And then we're also, of course, working on the paper with myself, Dick, and Leylith on the syncytium. And so those are the two that are kind of coming out of that. So yeah, and then we have, of course, Susan and I have been working on the tensegrity stuff, and I don't think there's anything else outstanding. And anyways, I'm going to be presenting this week at the Open Room General Meeting. It's held on Zoom every year. Usually it's an update from everyone on what's happening in the group over the past year. So I'm looking forward to seeing what other people are doing. I've got actually our annual meeting posted on the YouTube channel already, so you can take a look at that. And it's just a recap of what we've been doing over the past year. But I'm gonna get some feedback there from different people at OpenWorm. They're usually pretty positive about things, so it's not like. But anyways, yeah. So I'll report back on that as well. So that's good. Other than that, we're kind of wrapping up the year. So if people have ideas for next year, we should get in touch and talk about what you'd like to do or what you'd like to see. This is a paper, as a bioRxiv paper on reconstructing Waddington's landscape from data. So this is James Briscoe, Eric Sigia, Dylan Cislo, and they're talking about Waddington's landscape. So just as a recap, Waddington's landscape is this developmental landscape, or sometimes you could think of it as a tree, where you can observe the the trajectory of developmental events from the one cell fertilized egg to a completed embryo. So every time there's like a cell division or a differentiation event, there's a branching, and the tree structure forms these canals or channels, and they use the term canalization to represent that process of channel making through that landscape. So it's kind of a you could call it a combinatorial tree if you'd like, but it's on this landscape and it carves something into that landscape. So it's not the same thing as like a gradient that we usually use in physics or we use in machine learning. It's a little bit different thing. In any case, those channels then are sort of the way in which you make an account of development. So you can look at the channels and their pattern, and that's important for things like buffering of phenotypes and other types of things that happen in development across embryos and across development. So they're going to, but one of the things about Waddington when he kind of came up with this concept was that it was largely, I guess, an analogy of like a landscape with channels, and that was the idea. There wasn't a lot of quantitative rigor to the landscape at all. So that's where this paper came in. Now we talked about a paper. We talked about a paper, I think about 2 years ago, maybe now. That was where they kind of did the same thing as what they're doing here. They tried to sort of quantify or quantitatively characterize the landscape with actual biological data, which you might think, well, that's ridiculous. We should have done that in the first place. But, a lot of times theoretical concepts have to be sort of, some sort of analogy, some sort of, you know, uh, sort of a diagram or some sort of thing that is generally applicable. And then you go back and you verify it with data. So, and that's what they're doing here. They're reconstructing a Waddington's landscape from data. So again, they mention that the development of a zygote into a functional organism requires that the single progenitor cell gives rise to numerous distinct cell types. Attempts to exhaustively tabulate the interactions within developmental signaling networks that coordinate these hierarchical cell fate transitions are difficult to interpret or fit to data. An alternative approach models the cellular decision-making process as a flow in an abstract landscape whose signal-dependent topography defines the possible developmental outcomes and transitions between them. So this is, they call it a signal-dependent topography. So depending on what's happening in the embryo, you get these, these channels, and then you get these branches in the channels where you can go in one direction or another. Prior applications of this formalism have built landscapes in low-dimensional spaces without explicit reference to gene expression. So this is where, again, people have kind of thought of this as a low-dimensional space. They don't talk about gene expression. They kind of talk about the embryo as is. You observe the embryo, you observe these branching events, and that's the description. What they want to do is graph gene expression onto that more explicitly. Like, what are the gene expression event or the changes in gene expression that happen that trigger these branching events in terms of different channels forming and branching off to different trajectories. So that's what they're going to do. And it's, of course, if you know anything about gene expression data, you know that's going to be quite challenging, because a lot of times we have to take gene expression data and calibrate it to some sort of phenotypic event. So, all these papers we see with these stains that they do, or these fluorescent images where they try to overlay things and show differences between that and a control, those are all attempts to sort of calibrate that to a phenotype, right? You see a gene expression difference or change, and you see the amount of change, and then you put that in anatomical context and say, oh yeah, this thing is changing in this location and at this time. And then that gives us sort of this, evidence that this is what's causing this. This is what's causing, or maybe not causing, but this is what's involved in this process. So you'll find 1,000 papers like that. But in this case, what they're trying to do is they're trying to really kind of work out this space, and that's going to be a little bit harder. So here we present a computational geometry framework for fitting dynamical landscapes directly to high-dimensional single-cell data. Our method models the time evolution of probability distributions in gene expression space, enabling landscape construction with minimal free parameters and precise characterization of dynamical features, including fixed points, unstable manifolds, basins of attraction. So what they're doing here is they're actually treating it like a dynamical system, and they're treating the Waddington's landscape like a dynamical systems diagram, where you have trajectories across that surface. We demonstrate the applicability of this framework to multicolor flow cytometry and RNA-seq data. So this is where we're getting different types of data that are going to be suitable for this approach. Applied to a stem cell system that models ventral neural tube patterning. So they pick a specific system here. We recover a family of morphogen-dependent landscapes whose valleys align with canonical neuroprogenitor types. So this is where they're looking at these valleys or these channels, and they're looking at these landscapes that are shaped by morphogen activity. So they're able to detect that through the RNA-seq data, and then they, and also with the flow cytometry data to look at the different types of progenitor cell. Remarkably simple linear interpolation between landscapes captures signaling dependence, and chaining landscapes together reveals irreversible behavior following transient morphogen exposure. Our method combines the interpretability of landscape models with a direct connection to data, providing a general framework for understanding and controlling developmental dynamics. So what I'm interested in here is to see what their diagrams look like, because that's going to be sort of the part of this paper, I think. So this is not, so they're not really modeling epigenetic landscapes per se. I mean, the way that Waddington originally envisioned them. But what they're doing is they're using that as a way to get to this question of what are the trajectories of development that are most common given what's happening during this biological process. So that's, and then of course, if you can understand that, you can understand the effects of perturbation. So if I have, like, say, for example, an embryo that is within this channel, if I perturb that developmental trajectory, it might go up and down the channel on either side. But if you do that, if you were like riding a bike down a canyon and you kind of went up and down the sides of the canyon, you would have to have some momentum to jump out of the canyon, or you'd kind of go back and forth along the bottom of the canyon. So it takes some, we could say energy, but that's not really the right term, to pop out of one of these channels and end up somewhere else in development. Basically, the phenotype is robust to changes in gene expression and changes in fluctuations in gene expression over time. So that's another reason why we want to understand these kind of landscapes, and then link that back to gene expression data, which we know has a lot of noise in it, or good intrinsic noise, where it's showing these fluctuations over time. All right, so this is an image of what they're doing relative to the Waddington landscape is B. A is sort of mapping from gene expression down to three-dimensional shape. And then C is this geometric model, which shows, and remember I said they were taking inspiration from dynamical systems. So these Waddington landscapes are actually like attractor basins in a geometric model. So let's look at A. So A is where we have what we call a GRN, or a gene regulatory network. And that's a network of genes that are being expressed, but they're not just being expressed, they're regulating each other in this network of interaction. So if you go in, a lot of times in papers, they'll go in and they'll look at different genes that they think are affecting one another. If you look at a developmental process, we know that one gene, if it's upregulated or downregulated, affects a downstream gene that's upregulated or downregulated. And so that gene A will have an effect on gene B. The activity of gene A will have an effect on the activity of gene B. And fortunately, when we have RNA-seq data, we have those data. We have the sequence, which we can look at and we can map to like a draft genome, and we can say this is this part of the gene that we think is responsible for regulating in A, that's responsible for regulating the thing in B, for which we also have a gene with a sequence. But we also have copy number of transcripts, so we know that we can look at its relative expression relative to a baseline, but also relative to one another. And so we can look at this process of regulation, upregulation and downregulation, as a consequence of some other thing in the gene regulatory network. So of course, why we're interested in gene regulatory networks instead of just genes being upregulated or downregulated, single genes, is because then we can look at spatial patterns being formed. So one of the things about reaction diffusion morphogenesis is we generally need to have a series of morphogens and model those as genes that are affecting the expression of one another. So in reaction diffusion morphogenesis, we have these gradients of a certain gene being expressed or gene product that has some distribution in space. And then when those distributions overlap, there's a sharp boundary that forms. And it forms these sharp boundaries that you see in the spatial pattern of spheres that are kind of have a sharp boundary with the yellow background. And so that's all sort of the action of these different morphogen gradients, which in real terms, in data terms, are going to be these different genes that are being expressed with a spatial decay or with some spatial distribution, and then the overlap of those distributions for different morphogens. And so that's where we get our patterns. But that's only useful in a two-dimensional case. And of course, biology isn't two-dimensional. We have those genes being expressed in 3 dimensions. So then we need a 3-dimensional shape to characterize that. That's true pattern formation morphogenesis. So that's what we have there. And then B, of course, is the Waddington landscape. So we use that as their analogy for thinking about if we have this ball as our starting point, we can have this branching event where it goes down 2 canals. So we start off at our initial point, which is the ball at the top, and it rolls downhill and it rolls down one of these 2 canals or channels that you see here. And so depending on the channel it rolls down, it has a certain set of developmental trajectories available to it. And the idea is that if you have enough branching events, you end up at some portion of the space that's restricted in terms of, you know, what's available to the embryo. So to put that in less slippery terms, you have an undifferentiated one cell, and then you have these differentiation events which specify the phenotype. And you end up in a place with a very specific phenotype. And when you have a very specific phenotype, you can't just suddenly change to another totally different phenotype. Now, this is interesting for a number of reasons. Of course, we talked about you know, how you have this developmental trajectory and how you have these division events. It's also important in cell differentiation. So if we talk about like different stem cells are different, like if we have a neural progenitor cell and it differentiates into some neural cell, like a neuron or glia or something like that, then that's the same process. We have these branching events, and you end up with this progenitor cell at the top, which is the ball, and at the bottom you have very specific phenotypes and cell types. And that involves a change in a number of genes in a network that as they change their expression, they lock the cell into these different fates. Now, we talked about this model of Plinko, which is a game from the old Price is Right show where you know, some people call it a Galton board, where you have a chip that goes down this set of um, you know, nails or something, and it bounces around between them, and it ends up kind of moving back and forth, and it ends up in a bin at the bottom of the board. So if you look up Plinko board, or if you look up Galton board on Google or something, you can find it, and you see what I mean. That it's basically you have an initial condition, you drop this thing, and it goes down through the board, through the different pathways, and it ends up in a specific place. In that case, you know, it's partially stochastic and partially dependent on your initial condition. In the Waddington landscape example, it's a little bit more complex and involves feedbacks to an existing genome and an existing pattern of differentiation in a lot of cases. And then finally, the C is the geometric model. So we're just using the Waddington landscape as a metaphor, but really what we are interested in are these series of attractor points or attractor basins. So our initial ball is here, and it kind of rolls around in this attractor basin, or it jumps to another attractor basin, and the dynamical system can transit between attractor basins. And depending on, again, these changes, they end up in a very specific attractor basin in a very specific position. So it looks like Susan is here. Hello? Pinball. Yeah, pinball is the... Glad you could make it, Susan. So you're doing a lot of dynamical systems here. They're also, working on a computational framework. So they have their algorithm here, where their goal is to build a method for simulating the high dimensional dynamics of a developing biological system that can fit directly to experimental data. The application developed here is to single cell data, but the formalism is broadly applicable to different types of developmental dynamics. So here's where they give some math for what they're doing here. So they want to have this representation of a time-dependent state of the cell, which is maybe operationalized as the expression levels of D different genes at time t. So it would be like one RNA-seq run, where you get like all these genes that you're sampling their expression at time t. You might do another RNA-seq run at a different time, and so each of those are different time-dependent states. We assume that the time evolution of the system is governed by the following Langevin equation, and that's this here. This is the dynamical system, basically. Here, the drift term V is assumed to be gradient-like for a given metric tensor G and potential U, which serves as a Lyapunov function in the Morse-Smalley system we are modeling. The stochastic term eta t, standard white noise with mean 0 and covariance given by this term. And so the indices run over the dimensions, and d is the positive diffusion constant. So that's just details of this equation here. The biological systems we model are typically not stationary, and there's no explicit link between U and equation 1 in any equilibrium distribution that exists. By design, G does not appear in equation 2, which they define here. I guess G is carried over from 1. It's not carried over to 2. Okay, so we denote by theta a vector of time-dependent parameters that control the topography of the dynamical landscape, and operationalize that's the expression levels of a set of morphogens at time t. So that's the dynamical landscape. And then as those morphogen levels change, the expression levels change over time, the dynamical landscape changes, and it gives you this landscape by which you can derive the trajectory of this dynamical system. Changing these parameters can destabilize existing states, introduce new states, or bias the system towards a particular outcome. We usually omit the explicit dependence on theta for the sake of notational simplicity. In the deterministic limit, the trajectories xt may evolve along a low-dimensional submanifold, but high-dimensional noise will generally blur this confinement. So the trajectories move along this sort of tight, I guess, like what I was saying before about something resonating in a canyon as you move, and you kind of move up and down the sides of the canyon. It's basically what they're saying here, that high dimensional noise can play a role in sort of blurring this trajectory, but basically we can work out the mean trajectory and be okay. In this work, we do not attempt to explicitly encode the geometry of M, and instead extend the stochastic dynamics to R of D by defining a function U that keeps trajectories close to M. So what they're doing here is they're just trying to like approximate this trajectory as it would be with the sort of mean behavior. It is not usually possible to continuously follow the state of a particular cell xt over time due to the destructive nature of experimental methods. So, we're basically killing the cells as we measure them, and so that's a problem. We can't go back to the same cell, measure it twice. So we need to find a distribution whose time evolution under equation 1 is given here by this differential equation, where delta denotes the Laplacian and RD. Standard finite difference or finite element methods for solving this equation are hopeless in high dimensions due to the exponential explosion of mesh elements needed for reasonable numerical accuracy. Well, that sounds like something Susan's familiar with in some way. So we propose to circumvent the exponential increase in computational resources with dimension by defining a Markov process restricted to representative points sampled from the data. The time of evolution of pxt is given by equation 3 and is equivalent to the following recursion relation for epsilon that's much smaller than 1. It's just here, this equation, where they look at the transition probability from state y at a certain time to state x at time t. In what follows, we adopt the simplified notation here, suppressing the dependence on time, which enters only through Vn for theta for mixed epsilon. More details are provided in the supplementary information. We're not going to get into that, but I just wanted to kind of think through this a little bit and look at the math and see what they're doing. The discrete representation for our underlying dynamical manifold is a set of endpoints sampled from the experimental data. These points will typically be sampled from all time points and all experimental conditions in order to achieve robust coverage of all relevant regions of the gene expression space. This is an interesting point because our gene expression space, on the one hand, is really limited to what we can measure, right? We can't say something about things we can't measure. That's why we want to take things at multiple time points, but even then we can't measure the time evolution of within a cell because we destroy the cell when we measure it using something like RNA-seq. We could get that information from another cell at another time point, but it's not really the same process, the same expression process. We can kind of only approximate it. And so this is our gene expression space, and we want to understand sort of the most relevant regions of that. So not all of what we characterize as gene expression space is going to be useful to us. Sometimes we get a lot of noise, which is, like I said, it's an intrinsic noise, so it's good noise, but it's also something we know that is kind of variable. So if we take the mean values, we're okay. And as well, we want to be able to know kind of where, at what time points, say, there's sort of abnormal fluctuations happening, and at what time points gene expression is sort of close to that mean. And so we don't want to, sample the gene expression space once and not end up with values that are basically outliers. So this is an important point. We have to kind of figure out what this gene expression space looks like. And it's not easy to do because we need to have the right type of data. We need to characterize the data before we can even approach this sort of enterprise of building a geometric representation of it. The local base density PB of the points in M hat will be approximated using kernel density estimation, which is KDE. Smoothing is sum of functions centered on the xi by a convolution with a Gaussian kernel with a specified bandwidth. So this KDE, of course, we use this in machine learning a lot to normalize data. Then they go through their choices for representing different types of relations between cells and that sort of thing. Figure 2, and we'll get to that, explores the application of this method to the so-called heteroclinic flip potential. And that's this uxy function here, which is this equation here. This is a crucial landscape that defines the common situation in development where a single progenitor cell type differentiates into one of two possible daughter cell types. The parameter A, which is here, controls the stability of the progenitor, while the parameter B, which is here, tilts the landscape vertically and controls which daughter state receives the output of the progenitor state. Flipping the sign of B, so right now it's positive and you could flip that to negative, results in a global bifurcation, since it entails the reconnection of the unstable manifold of the saddle nearest to the progenitor from one daughter state to the other. So basically what they're doing is they're building this function here, and they are trying to figure out what the shape of this landscape is, but they're also trying to figure out sort of how to connect them as there's this split in what was originally a single state. So why don't we look at figure 2? So this is a discrete path integral formulation that solves the Fokker-Planck equation on non-uniform point sets. So A is the heteroclinic flip potential. The magenta points are stable attractors. The cyan X's are saddle points, and the orange curves are unstable manifolds. So these are the magenta points here. We have our orange trajectories, these curves, these are unstable manifolds, and then our purple curves are the stable manifolds. So the purple curves come down like this. These are the stable manifolds, and these orange ones are the unstable manifolds. And then the magenta points are the stable attractors along those unstable curves. So this is all in this sort of dynamical system space. So that's a little hard to understand because there's so many lines. But basically you have this transition from one attractor basin to the other. And so that's what makes these orange trajectories unstable. You're moving from one stable state to another, and in between you get this bistable state where things can go in any direction. So you have these stable states within the attractor basins. But then you also have these stable trajectories, or they call stable manifolds, that kind of cross these bistable points between the attractors. So that's what they're showing here. Now for B, that's where the discrete representation of M hat of the dynamic manifold is sampled from a 3-component Gaussian mixture with one isotropic component with a covariance of 0.25 centered on each of the potential minima shown in A. So this is again showing these stable points, showing these attractor points, these attractor basins, and then showing these regions in which they exist. So you can see this is another sort of way of representing it. C is where we have the terminal density at time 12.5 computed using equation 6 and an initial distribution centered around the progenitor state, which is here. So this is just showing at a certain time what this looks like, and this just shows the landscape with the attractor basins and the stable points. And then presumably this would change over time. And then D is Kullback-Leibler divergences between the finite difference probabilities, PFD, and the path integral probabilities, PPI, normalized by the entropies, HFD, of the finite difference solutions of the Fokker-Planck equation. Finite difference solutions are transferred onto the non-uniform point set by area-weighted averaging for direct comparison. So D are the Kullback-Leibler divergences, just showing this for DKL over time. And this just shows how this operates. So there's this initial point here at 0 time, and then it goes up when it splits, and then it converges down to a value, I guess a moderate value like 5 once it goes off. So the trajectory splitting, so you see that at time 0, it's 0. But then after it splits, there's this high, sort of this rise in this DKL parameter, and then it settles down after it's split. So that's using, of course, Kullback-Leibler divergence. All right, so that's enough of that figure. This is figure 3. This is the fitting the dynamical structure of a synthetic data set. So this is where they have a synthetic data set. They're showing a number of things in this. So A is up here where you have, first of all, you have this time series where you have these experimental conditions theta. These are time slices here. These are this is the theta dimension. This is time. So you have these different forms of data. Then you have these point clouds that are converted to probability densities on MAT. So this is taking the data that we're collecting from this experiment at the top in A, and then we're looking at C. We're plotting this out as basically a series of distributions, probability densities. And so you can see that, there's a strong sort of set of points here that shows us a stable state here with some points out here. And then the point cloud evolves to include these other states out here, and you can start to see those emerge in the probability densities. And then B, where is where we have this dynamical manifold M hat, which is represented by points subsampled from all data sets. So this is where we can really turn these probability densities into these dynamical systems landscapes, where we have our density maxima, which are stable points in the dynamical system. We have our inferred saddle points, and the saddle points just refer to a bistable point, or the point at which you cross from one valley to the other, or from one attractor basin to the other. So you go up the edge of the attractor basin, you get to the top, and on the other side is another attractor basin. So it's like going up to the edge, going over the edge, and down into the next one. And those saddle points are bistable, meaning you could go backwards, back into the original attractor basin. You could go forwards into the next attractor basin, or you could sit there with a bistable potential. And the reason I mentioned that is because that's one of the things that happens in differentiating cells, or cells that are reprogrammed from one somatic state to another somatic state. They go from that somatic state, they get reprogrammed to this bistable state, which is where they express markers of stemness, but they are also expressing markers of another cell fate. And so they sit at the saddle point for a while, and then they go back into another attractor basin, a second attractor basin, which represents the target phenotype that people want to reprogram the cell to. This is also true in cell differentiation from progenitor cells. You get these saddles where they go from one cell state to another. And then in D, we have, let's see, in D, we have a schematic of our interpolation procedure used to construct a smooth potential for a given set of fixed point heights hi, defined on the sinks and saddles. U linearly interpolates between hi star along the unstable manifolds and smoothly interpolates everywhere else by minimizing the energy. And that's it from equation 8, which penalizes curvature. So they're using this energy minimization scheme, which basically allows us to interpolate unstable and unsmooth manifolds and make them become smoother as a representation. Okay, so that's basically what they're doing here. The fit parameters can be iteratively optimized until we have a dynamical landscape that accurately captures the dynamics observed in the data. So we end up with this sort of rough approximation of a landscape. Then through energy minimization, we try to make it a smoother landscape, and then we can iterate those parameters additionally so that we can get a dynamical landscape that matches the actual data that we have. So again, we're constructing this maybe from a single data set, or from some sort of probability distribution. We're then smoothing it out, and then we're fitting it to different different sources of data to get this landscape that's accurate. And then E is here, where we have our base potential. This is additive with the interpolated potential, and this gives us our dynamical potential. So again, we're interpolating values from our base, so we're doing maybe our observation of states from our data set that we have. We might have a single RNA-seq data set. Then we have an interpolated potential, which is a smoothing process, and then we have the dynamical potential, which describes a dynamical system and its possibility space. So we can model these trajectories as a means of something that fits the data, but also something that generalizes across data sets. Okay, so they do use some simulated data in this just to calibrate things. Then they get into landscape model for neural tube patterning. This is where they kind of show this for a specific model system. And that's, I'm not going to get too much into that. It just talks about sort of thinking about this genetic regulatory network with respect to specific genes. So, in the neural tube, we have opposing gradients of BMP and Wnt from the dorsal side and sonic hedgehog from the ventral side. So now we're adding in this anatomical specificity. We have these different gradients in different parts of the anatomy. The two morphogen gradients define 11 domains of neural progenitors, range linearly along the dorsal ventral axis, each of which is uniquely identifiable by a characteristic pattern of transcription factor expression. So this is where our RNA-seq data comes into play. We know in each cell we're going to have some characteristic expression. This is going to be spatially restricted, meaning that each cell, depending on its position in the anatomy, is going to have a specific, we should expect a specific profile in terms of gene expression. And then we can look at that and we can construct this dynamical system from looking at those data, putting this together into a manifold or a series of manifolds, and then smoothing it out, and then generalizing it to other. This figure 5 is dynamical manifold architecture of in vitro proneural patterning. So this just shows some of the patterning in the system here. And this shows the basins of attraction for different cells. And that's how they're doing this. They're plotting this out as a set of points in a manifold. Then the discussion is where they talk about this method applied to an MESC model of ventral neural tube patterning. Our method produced a family of dynamical potentials as minima aligned with the canonical vertical neural tube progenitor types linked by codimension 1 saddles. Changes in morphogen concentration sequentially create and annihilate stable states, matching the cascade of observed fate bifurcations in vivo. States appear to be arranged around a loop in the 5D state space. Our algorithms discretize the path integral form of the Fokker-Planck equation for gradient systems at additive noise. So the Fokker-Planck equation, of course, is continuous. They're discretizing it and trying to find a way to apply it to the data sets that they have. This formulation naturally captures the morphogen dependence of the dynamics through a one-parameter linear interpolation between two dynamic potentials. And so they don't, another rival approach is where they separate Hough functions that had to be fit to each of the parameters in the interpolating polynomials. So they're using this polynomial function as a way to sort of come up with these dynamic potentials. And there are different ways to do this, and they're just kind of pairing it with the citation 10. Even assuming an underlying MS structure, the result in figure 6 is a remarkable simplification and strongly suggests a gene epistasis can arise from minimal interactions of pathway components filtered through nonlinear geometry of a developmental landscape, as previously shown in this reference 6. So they kind of go back to the idea of canalization, and they say the ability to linearly interpolate potentials is another manifestation of canalization. So canalization, again, is where you keep that phenotype within those channels. You can have, you might think, well, if gene expression is so volatile, which it is, why don't developmental trajectories just drift off in different directions all the time? And the answer is because they exist in these channels. And the channels aren't just theoretical, they're just the product of epistasis or the product of these gene expression networks that are keeping the entire system stable and within the bounds of these attractor basins or channels. And so even though they branch off into different directions and specify their phenotype, they also exist in these channels that buffer the phenotype, keep it sort of within a set of operating parameters until there are these differentiation events. So that's kind of what they're getting at here. Quantitative models are valuable not only when they accurately reproduce experimental data, but also when their predictions deviate from observations in informative ways. And so this is just kind of talking a little bit about that. And that's the paper. I think that's a pretty good paper. I like the kind of the way they build the mathematical model, and they're really kind of getting at this problem. Although I still think it's kind of um, maybe hard to connect the RNA-seq data to the phenotype. Well, I mean, in terms of the branching events, so we could make it compatible with the differentiation and tree approach that way. I'm not really sure. I think there are a couple missing steps, obviously. I guess that's the conceptual model, because they're not constructing a Waddington landscape. They're constructing a dynamical system that sort of has the analogy of the Waddington landscape. And it's like, this is what that would look like if we did use the dynamical model instead, or more of a dynamical model, and then kind of mapping those components to the landscape. So it's yeah, I don't know if they need to necessarily use the Waddington's landscape as an analogy here. I mean, it makes the comparison a little bit more understandable, but it's not the same thing. All right, so I think that's it for today. I don't know if I'll see you before the new year, but if not, happy new year. Okay. All right, thanks. Bye. So to tie up loose ends with the dynamical systems work in the other paper, I wanted to talk about one more paper. In this case, we're talking about something called Arnold tongue entrainment. And this is another dynamical systems paper, this time focused on the embryonic segmentation clock. So we're focused on yet another dynamical system in a different developmental model system. So this is in the journal eLife. So let's go over the abstract and then get a little bit into what their take on dynamical systems applied to development is. So living systems exhibit an unmatched complexity due to countless entangled interactions across scales. Here we aim to understand a complex system, that is segmentation, timing, and mouse embryos, without a reference to these detailed interactions. To this end, we develop a coarse-grained approach in which theory guides the experimental identification of the segmentation clock entrainment responses. So we're dealing with this clock, something that is a regular process, looking at the different parameters and trying to extrude them from experimental findings. So they're trying to quantify all this. This allows them to derive what they call a phase response curve and a construct called Arnold's toungues, and we'll talk about those are in a little bit. So they get those for the segmentation clock, which reveals its essential dynamical properties. Our results indicate that the somite segmentation clock has characteristics reminiscent of a highly nonlinear oscillator, close to an infinite period bifurcation, and suggests the presence of long-term feedbacks. Combined, this coarse-grained theoretical experimental approach reveals how we can derive simple, essential features of a highly complex dynamical system, providing precise experimental control over the pace and rhythm of the somite segmentation clock. So they're trying to take this construct, the segmentation clock, build a model from the data, and they're able to derive these different higher order constructs, the phase response curve, and Arnold's toungues. So they asked the question, how do we gain insight into a complex system which exhibits emergent properties that reflect the integration of entangled interactions and feedback regulation? And so they actually cite David Marr and Tommaso Poggio, who are computational neuroscientists. Marr and Poggio in 1976, where they want to understand the complexity encountered when studying the nervous system or a developing embryo, and they say that that requires an analysis at multiple levels of organization. So they're actually bringing the Marr's three levels into this conversation, which I find interesting. Their core tenet is that also in biological systems, different levels of organization while obviously causally linked, exhibit only a loose connection, and importantly, can be studied and understood independently from each other. So, this is not just something that you see in biology. You see this in physics, where renormalization techniques allow us to coarse grain systems and obtain scale-free theories, allowing us to define universality classes independent of the precise details of interactions. And so there's citations here, Carty and Bialik, 2015. Another recent example is the parameter space compression theory, showing how complex systems in biology or physics can be typically reduced to simpler descriptions with few parameters. Going one step further, this suggests that one might be able to study and control complex systems, provided we identify the essential macro-level behavior. This is possible because only a limited number of universal descriptions exist, with defining behaviors and properties that do not depend on the detailed implementation. A central challenge that remains is to implement these theoretical ideas to the experimental study of biological complexity. So here they develop a coarse-grained approach, combining theory and experiments, as we did in the last paper, to study a cellular oscillator ensemble that constitutes the embryonic somite segmentation clock. Functionally, this clock controls the periodic formation of somites. We've talked about this in a previous meeting. You get these somites that form on a regular basis, and that's like a mechanism for producing eggs. It controls the periodic formation of somites, the precursors of the vertebral column, and other tissues. So this is where you form this structure sort of on a regular basis. Molecularly, the segmentation clock comprises the oscillatory activity of several major signaling pathways, such as Notch, Wnt, and FGF signaling pathways, which show oscillatory demands with period matching somite formation that is around 2 hours in mouse embryos. So there's this clock again. This is different than the cell division machinery and the cell division clock. This is where you sort of have these oscillating genes, and everything has to kind of be coordinated across, with gene expression and cell division to make this happen. More recently, segmentation clock oscillations of the period of around 5 hours has been identified in human-induced pluripotent stem cells, differentiated into paraxial mesoderm, identifying a set of around 200 oscillating genes, including targets of Notch and Wnt signaling. Adding to this complexity, these periodic spatiotemporal wave patterns are linked to an underlying spatial period gradient along the embryonic axis that is signaling dynamics in cells close to the posterior of the embryo, which oscillate faster compared to those in cells located more anteriorly. So there's this, again, this anatomical difference in the embryo. This is a spatiotemporal sort of specific signal. So, it's very similar to what you're seeing in the other paper with respect to pattern formation. Such a period gradient linked to the segmentation clock has been identified in several species, as well as in vitro assays culturing intact or even dissociated pre-Semitic mesoderm, or PSM. Of note, an analogous oscillatory system was also described during segmentation on arthropods, and while distinct at the molecular level, it also exhibits spatiotemporal wave patterns traversing the embryo axis, again with an indication of period gradient. So moving up and down the axis, the anterior, posterior axis, and setting up these things as it moves. And so we see these waves again and again, anatomically based waves. And so, again, this is something that is also analogous to differentiation waves, and so it's an interesting kind of point that they're making here. In this work, we coarse-grain these underlying complexities in dynamical systems, macro perspective on the segmentation clock, studying it as a single-phase oscillator. We build on the theory of synchronization and entrainment to first perform a systematic experimental characterization in response to perturbation. In turn, these experimental quantifications allow us to derive phase response curve that uniquely characterizes the dynamical properties of the segmentation clock. This new insight provides means to understand and control the timing of a complex embryological patterning process. So this is figure 1, and we see this E10.5 mouse embryos is just the developmental period in days, where they're cutting off part of the tail. They have the tail here, and they're doing this live imaging of the tail bud, and they're putting these into the system, and they're perturbing the tail bud, and they're looking at the response. So B through D is the characterization of the phase space. We have different trajectories in how they move. So you have the cycle that can diverge. In B, you can have the cycle that converges in C, and then you have the dynamics of entrainment in D, where you either have something that's phase or anti-phase, and it depends on the height and the period of the sine wave here. So here we have two sine waves that are overlapping partially, and we see this delta theta, which is this difference between the two oscillations. And so that can show you how out of phase something is, or how in phase something is. And so when it's in phase or out of phase, it has these dynamics where it deviates from this cycle. So this is, and we're going to talk about what Arnold's tongue is in a bit, but this is the Zeitgeber period versus the Zeitgeber strength. And so for purposes of thinking about what this is, we're not going to kind of worry about what Zeitgeber periods and Zeitgeber strength are, but just know that Arnold's tongues are basically these regions of a period so like for a certain Zeitgeber period, you start at the bottom for, sort of a strength of 0, and then you have this 1-to-1 entrainment and a 2-to-1 entrainment. And so if you look at the graph, the Arnold's tongue, they call it tongue because it kind of goes down to the bottom, and it's this tongue. It's kind of a cheeky characterization of dynamical systems, but basically you start with a Zeitgeber strength of 0, and the Zeitgeber period is a point, and then it diverges from that point as the strength increases. So the 1-to-1 entrainment is wider at the top, the 2-to-1 entrainment is wider at the top, and they overlap at some point of great strength. In this figure E, they have 3 points that they sample, F, G, and H, which are these F, G, and H insets. So now we have, so the Zeitgeber period is TZEIT, and that is contrasted with TOSC, which is this point here. And you can see that, like, where the TOSC is greater than the TZEIT, you have this pattern in F, where TZEIT is greater than TOSC, you have this pattern here, where it's kind of a longer period, and then H, which is where the TOSC is twofold to TZEIT, then you have these even broader periods here. So if you're familiar with circadian oscillations, this is sort of the terminology they use to understand that. The Zeitgeber strength and Zeitgeber period involve this sort of entrainment of an external stimulus onto an internal rhythm. And so this is where they're able to perturb this internal rhythm by entraining it to an external source. And so that's kind of what they're doing here. And they're showing these different, like the TZEIT is in the fuchsia color, and that just is overlain over this sine wave. And so you can see the results here. Okay, so why don't we get into this theory of synchronization guides and the experimental study of segmentation clock entrainment. So their experimental study is based on and guided by the theory of entrainment of oscillators by an external periodic signal, which is what we just said, a subset of the general theory of synchronization. So entrainment is observed in an autonomous oscillator, adapts its behavior to lock to an external periodic signal, and this is the thing they call a Zeitgeber, so it could be light, it could be a signal that affects gene expression, etc. The general theoretical framework To understand entrainment requires the definition of oscillator phases and their response to perturbation. We have to understand how a certain perturbation affects the endogenous signal and how that affects entrainment. Assuming the Zeitgeber consists in periodic pulses, entrainment is observed when the phase of the oscillator of the time of the Zeitgeber is constant, technically a fixed point on a Poincaré return map, which is a tool that we won't get into, but just let's say that basically you have this fixed point, this zero-dimensional point. This defines period locking, also termed mode locking. Entrainment is not only always manifested, and conditions for its existence can be derived. Quantitatively, when entrainment occurs, the Zeitgeber induces a periodic phase perturbation or response of the entrained oscillator, which exactly compensates the detoning for a period mismatch, which is T-Zeit versus T-Osc, between the Zeitgeber and the free-running oscillator. So the free-running oscillator is the endogenous sine wave, the endogenous process, and the Zeitgeber is the thing that's entraining that, the external signal. For this reason, when the detuning is very small, a weak external perturbation is enough to entrain an oscillator. Conversely, if the detuning is big, a strong signal and associated response is required for entrainment. One can then plot the minimal strength of the Zeitgeber versus the corresponding detuning. These maps are more commonly known as Arnold's toungues. So Arnold's toungues predict the period and phase locking behavior on oscillatory systems. It is different as electrical circuits, oscillatory chemical reactions, or living systems such as circadian rhythms. We can observe more complex patterns of entrainment. So for instance, stable phase relationships can be established, where the entrained oscillator goes through n cycles for every m cycles of the external signal, defining m-to-n period locking. So this is where we have a certain number of cycles of the endogenous process for every m cycle of the external signal. So you might have 2 cycles and then it's entrained by the external signal. And then that ends up being kind of a thing that, as those things become entrained, you get changes in the endogenous process and you end up with this effect on the dynamical system. In this case, the instantaneous period of the oscillator matches m to n, m over n, the period of the Zeitgeber. Corresponding Arnold's toungues can be obtained, leading to a rich structure for entrainment in parameter space. And so they talk about this periodic activation of notch signaling by a heat shock-driven expression of the ligand delta as a model of this in zebrafish. So you have the notch signaling activation, which is the endogenous signal. You have heat shock-driven expression of delta, and so delta is driven by an external signal, or the expression of delta, and then that is affecting this periodic activation of notch signaling. So you have this gene regulatory network where you have delta, you have notch, delta is affecting notch, delta has its own period, notch has its own period, and delta and affecting notch synchronizes the two, and so they become phase locked. In the cited study, the readout to assess the effect of the periodic perturbation relied on the somite size and morphology. Well, this gave important insight into how morphological segmentation, the PSM and somite length, are affected by a Zeitgeber. Experimental investigation on the underlying signaling dynamics upon periodic perturbation remains limited. So in this experiment, or in this paper, they use a microfluidics-based entrainment setup, and they show that using a quasi-2-dimensional in vitro segmentation assay, they can recapitulate segmentation clock dynamics and PSM patterning. They're able to take control of notch and wind signaling oscillations, and as they can control those different oscillations, they can look at this entrainment process and then derive this mathematical model. So if you read about Arnold's toungues, it's a little confusing. They use a specific way of thinking about Arnold's toungues in this study, but more generally in the study of dynamical systems, Arnold's toungues are this sort of pictorial phenomena of fractals and other things. So sometimes you'll read about it as sort of this very specific definition in dynamical in looking at these entrainment of different signals, but it also has applicability to to chaotic systems. So Vladimir Arnold was the person who this is named after. Vladimir Arnold was a Soviet and Russian mathematician, and did a lot of work in geometric theory, dynamical systems, and algebra. So Arnold's tongues are a pictorial phenomena that occur when visualizing how the rotation number of a dynamical system or other related invariant properties change according to two or more of its parameters. So this is capturing multiple parameters, looking at the rotation number. So in the case of our signals, or entrainment of our signals, when we think about a sine wave, we're thinking about rotation. That's what we do when we do That's what we do when we do a Fourier decomposition. We think in terms of a circle, or rotating around a circle, or rotating around 360 degrees of arc. And so this is where we're getting this from. So the regions of constant rotation number have been observed for some dynamical systems to form geometric shapes that resemble tongues, in which case they are called Arnold's tongues. So this is rotation numbers for different values of two parameters on a circle map here, and you can see these are Arnold's tongues here in red, and they form these kind of things that protrude out, and they call them tongues for whatever reason. Arnold's tongues are observed in a large variety of natural phenomena that involve oscillating quantities, such as concentrations of enzymes and substrates, and cardiac electric waves. Sometimes the frequency of oscillation depends on, or is constrained in the form of phase locking or mode locking, to some quantity, and is often of interest to study this relation. So, for instance, the outset of a tumor triggers in the area of a series of substance, some sort of protein, that oscillates in its expression, and those interact with each other. So when you have something like a tumor, you get a bunch of different types of proteins. Their production oscillates over time, and they interact with each other. And then we can look at those interactions using Arnold's tongues. So if we're talking about gene expression networks usually have some sort of set of constituents that are outputs. But it's not just the output that we're interested in. We're interested in the internal process. So we might have two genes, for example, that interact through some sort of interact through epistasis that lead to the expression of another gene. And so that's something we might want to understand using Arnold's tongues, especially if it has this property of fluctuation or of oscillation. We also want to understand the output and how that interacts with other outputs of other gene regulatory networks. So we're kind of playing on the complexity of systems where we have a bunch of different variables and how they're interacting, especially when they're doing this at different periods of oscillation and entraining each other and things like that. So in the case of the tumors, where we have simulations that show that these interactions cause Arnold's tongues to appear, the frequency of some oscillations constrain the others, and this can be used to control tumor growth. So we can understand how these oscillations get entrained, how they affect one another. We can use that to control the attain the complexity of that system. Other examples where Arnold's tongues can be found include the inharmonicity of musical instruments, orbital resonance, and tidal locking of orbital moons, and other types of phase lock loops, and other types of electronic oscillators such as cardiac rhythms, heart arrhythmias, and cell cycle. Cell cycle is particularly interesting because, as I said, that is also another oscillatory process that gets coordinated by the expression of multitude genes, and it's variable by cell type and other things. So the length of a cell cycle is, and I'm not run across the paper for these Arnold's tongues look at cell cycle. I would add here that people have also used this to look at sensory motor coordination. J.A.S. Kelso has done a lot of work on this, where they use synergetics to look at sensory motor coordination, and they've derived these Arnold's tongues from those data. to show how muscle synergies sort of coordinate motor control and all these other things. So it's an interesting set of results. One of the simplest physical models that exhibits mode locking consists of two rotating discs connected by a weak spring. One disc is allowed to spin freely, and the other disc is driven by a motor. So you have this driving frequency, and you have this other disc, which is, we're going to entrain that to the motor-driven disc. Mode locking occurs when the freely spinning disc turns at a frequency that is a rational multiple of that driven rotator. So this is where we have this, this resonant free, or this mode locked frequency that is translatable to what the motor driven one is. So it's like if you had a gear shift where you had like a motor driven wheel, and then it's connected by a chain or a belt to a non-driven wheel, and those things have to be synchronized in order for the mechanical system to work. The simplest mathematical model that exhibits mode locking is the circle map, which attempts to capture the motion of spinning discs at discrete time intervals. So when you look at an Arnold's tongues, you can look at this bifurcation diagram, which has black regions which correspond to these Arnold's tongues. And so like a lot of these kind of diagrams of chaos and complexity theory, you have to stare at it for a while to really appreciate what's going on, because there's so much that they visualize. Basically, these black lines are the Arnold's tongues. Those are frequency domains that are related to this phase locking. And so there's a whole bunch of mathematics underlying it. You build the circle map, because remember, we're talking about these circular relationships, or these curved relationships, these relationships of arc. And so that's important to understand. And so we have that, and we have families of circle maps which become endomorphisms of the circle to itself. And so we can relate other phenomena to the Arnold's tongues. We can understand the natural frequency, the periodic function of things, and then we can understand the coupling strength between two oscillators.